Okay, so now I'm going to talk about data lifecycle and standards and best practices. So this will reflect on some of the things I mentioned in the first bit. So this really comes out of a paper that's been accepted for GigaScience that's been part of the Elixir Accelerate Consortium. So there's multiple authors from 9 different institutes, and the paper really covers the whole process from that process of description of the sample, description of the sequence, description of the analysis, archiving of the analysis, this whole the whole pipeline all the way through from essentially the environment to something that's going to go into the database. And this is really refers to metagenomes, but what the points that we raise in this paper is equally applicable to metatranscriptomics, metaproteomics, and it really highlights where we already have some standards, but also where there are gaps. So really, for your data to have any form of longevity to the scientific community is you've got to archive that sequence data with contextual metadata. So what does that exactly mean? So here we really want to know, you know, the European Nucleotide has spent a lot of time trying to capture this information. So we want to know how it was sampled, how it was extracted, how it was stored, what sequencing platform. Where did that sample come from? What were the environmental conditions? Lat long, depth, pH, salinity, temperature. What are the clinical observations? Was it a patient? Was it control? So this all sounds fairly obvious, but you'll be amazed that how many samples just lack this sort of information that get deposited. So that is partly why we've teamed up with the ENAs. So as I mentioned, we broker everything that we have in EBI Metagenomics through to the ENA. So they very much have set up the set of tools to capture the person who's doing the submission, to capture all of that sort of when, where, what data and the sequencing platform. And so they have a whole bunch of tools. Most people use the web-based tools for accessing the ENA, but there's a whole bunch of checklists. So when you go through a submission process, it's really important to identify the correct checklist and then start setting up that checklist of how to. You've got a metagenome, which environment, and it starts driving you to the most specific. Now, this is always a bone of contention for scientists, because usually when they're coming to submit data, they're usually finishing a job, trying to write up a thesis, or just wanting to get the paper out. And a lot of this falls by the wayside, and they don't actually collect it at the point of reference. So one of the things that we're really trying to drive is the need for people to collect this data at the point and be prepared and be aware of the questions that are going to be asked early, such that they capture the information at the point of time rather than trying to retrospectively identify this information. So why do you care about this metadata? So a very quick search of the literature will quickly tell you that, you know, samples that are stored at sort of minus 80, they can change. But if you don't freeze the samples straight away, there's really significant changes in the microbial community. So there's huge numbers of papers that describe the fact that if you capture a sample, unless you can immediately freeze it, you're going to be in all sorts of trouble, because that microbiota is going to change. However, how you freeze it also has key issues, in the fact that there are lots of people who say, I've found that the cryoprotectant you use when you're freezing really matters. So if you freeze without glycerol, you lose virtually all your bacteroidetes. Yet that same process seems to have virtually no effect on your archaeal component. So capturing all of this metadata is really key about how things were preserved. The DNA extraction is now recognized as probably one of the biggest factors. I'm not going to go through each one of these steps, but essentially however you extract the DNA is going to affect your sample. So if you just use traditional sort of lysis, that's not going to give you the whole representation of that community. This bead beating seems to be one of the most important things in terms of, you know, what you get to actually capture all of the component. And it also differs for eukaryotic organisms and viruses. So one of the things, though, we're not trying to, when we talk about standards and best practices, we're not trying to say everyone needs to use the same approach, but what we do have is the need to capture how you've actually done things. So this happens a lot. So we've got data sets where people have been looking at sort of DNA extraction kit, and we've captured the metadata and we've analyzed it, and if we're just going to throw all of the results from this tab into something like Megan for just visualizing. And here what you've got is you've got a comparison of two kits, kit 1 and kit 2. So kit 1 was just a traditional lysis method. Kit 2 had some chemical extraction associated with it as well. And because we've captured the metadata, I know that this first column and this first column here are the same sample. They're where it's not perfectly deposited, but at least I've got the metadata to know that these actually were the same sample. And what you're seeing is, as you compare each column, so 1 to 7 and 1 to 7 of kit 1 and 2, you see quite some substantial differences of cases where things are picked up where the other one doesn't. And so we see this as a real factor, and this is partly why we need to know how samples were collected. Sequencing. We also need to know the style of sequencing. This again sounds obvious, but the number of times that things that are labeled Amplicon versus shotgun and mislabeled, the number of samples that say they're shotgun, it's actually Amplicon, is quite shocking, and we find that just from the analysis. But what happens and what you do and what analysis you want to run is very different with those two forms or approaches to metagenomic analysis. The sequencing platform makes a huge difference. The length of reads and what you expect, and even if you just say take Illumina, is it paired or singled end? Have you pre-merged the mate pairs? All of this really matters and is absolute key to the downstream analysis. And if you want to use other people's data, you need to know this. So as an example of sequencing, so if we look at our QC plots, and here we've got a typical Amplicon data set, and what we've got here is the distribution, and what you expect is a bias. And that's, as you're going over a certain region, you expect some conservation and some variability, but you see that bias. Whereas shotgun data sets, you expect on the right hand side a fairly equal distribution between A, T, C and G. What you also sometimes see is that people have not sort of trimmed the adapter sequences off, and you get a skew here. So it's looking at the QC is really important, but we need to capture all of this information. We've done analyses across different biomes to see if our analysis makes if there's any sort of skew in terms of the analysis to how much functional information we can say about different biomes or sequences coming from different technologies. And really the take-home message is really it's all about the same, except possibly the engineered data sets where we can annotate more. And the reason there is that a lot of those engineered data sets are using organisms that are well known. So there's things like cheese fermentation comes out here. The key thing is that there's huge variance across all of these analyses. So the mean is always around about a third of the data, but the variance is absolutely huge. Knowing what primers you used is really key. So there's a really well-known example. So SAR 11 is a clade of bacteria, black plankton. They should make up about 1/3 of most marine data sets. However, due to the primers that were used a while back, this V4 region of the SAR 11 was just completely missed. And so it's absolutely key we know what primers are used, because that will skew your results. You can also, by using different approaches, you can find novel organisms in these data sets. And you know, until you've got all of this information, can you actually start believing that some of these are real? This is one of the Kryptonia from DOE, but you know, where they've found novel organisms, novel phylum, but they've got lots of metadata where we can really validate the fact that they initially started seeing this 16S and it was definitely real. They went on to sequence the genome. So I've already asked this question: can you recreate analysis in papers? And I think most people probably would say no, it's hard to. I've shown you this example of R2 pipelines, so I'm showing you the pipelines, but I know that it will be virtually impossible for anyone to recreate these pipelines unless they were very familiar with the tools and what we were already doing. And also spotting the difference between these takes people a while to do, and that's not really what we want to have. So command line parameters for all these tools are quite interesting. So I'm going to take one tool as an example. So these are all the command line tools for Hammer. Now I know this tool pretty well. So if I changed any of these options, I know that this would have absolutely no effect to my results because these are just outputs. So if I started playing with these thresholds, these might well start having some sort of effect, because these are about reporting inclusion thresholds. Then these are the gathering thresholds. Now these thresholds here I can tweak, and some of these I can tweak based on speed, but can I make any difference based, will they make any difference to the overall result? It's hard to say, but these are all things that I know about, and rarely do you ever see the actual full set of parameters that anyone has actually used reported in a set of analyses. So what's the difference between these corona views? So what does anyone reckon they know the difference between these three plots? So that here you've got your standard sort of corona view with bacteria in the middle going out. Are they different samples? Do people think these are different samples? Just eyeballing it. I'm not expecting you to see the names, but eyeballing those corona pots. Do you think they're different samples? Anyone? No? They all you think they're the same sample? So if you think it's the same sample, why do you think they're different? Yeah, so there's some big differences. So that, I mean, the number of concentric circles are different in terms of each one of these. So these are all the same sample. These are all the same input. All I've done is change the reference database I've used with Chime. That's the only difference. And it's fundamental. And I haven't even put here the version of the different databases I've used. And this is one of the key things for the analysis results. They all give you slightly different answers, and there's lots of reasons, and I can't go into all of those now. Here's another example. So that one was previously on an Amplicon data set that was a brown bear microbiome. Here we've got ocean samples, Silva versus RDP. They've got very different levels of cyanobacteria. Silva pulls out archaea. RDP doesn't. That's because RDP doesn't actually have archaea in the database, and they have a very different view about the taxonomy of cyanobacteria. So one of the things we're advocating is the use of ways of capturing all of these parameters. There are lots of different workflow languages. One of the ones that we're working on and looking at really adopting now in earnest is this common workflow language. What that allows you to do is describe each tool, and then you can chain those tools together. I'm just showing you an example of one. So you have all of those output parameters, or those parameters like I showed on that HMM search help, encapsulated in a document, and then you've got an associated document that actually defines those parameters where they're tied, and that allows you to recreate a command line, and then you can chain all of these together to re-describe your pipeline. So another wonderful joy that we always have is sort of comparing between experiments, and that becomes really, really tricky. So we allow a comparison within the EBI Metagenomics, and you're only allowed to compare the results from within the same project. The reason is that when you start going across different projects, they've all been sequenced differently, they've had different extraction systems, we've already started talking about that, but they've potentially sequenced at very different levels. So here's some estimates. So if you take, sorry, the quality of the graphic's not great, but the point is the same. So if you take how much of the community you've captured with a given amount of sequencing effort, so say you've only got enough money for 1 gigabases worth of sequencing. If you've got a human sample, sort of these 1 to 4, you've probably captured about 95% of that community. However, if you're looking at some soil samples, you're probably only about 20 to 30% of the community at best. With aquatic environments, that can range massively depending on that aquatic environment. You might capture up to sort of 50-60%, or it could be little as 20. So comparing data sets is really, really tricky, and rarefaction is also not a good thing, although it's widely done, because you're throwing away huge amounts of data. It doesn't actually deal with biases that are actually in the data for real. And so when you're doing the analysis, you've got to think, and when you want to do the comparing, you've got to think about, is it a fair comparison? And when those comparisons are drawn and any corrections, you've got to accurately describe those. So another thing in terms of where we're sort of getting to now is what should be kept from the analysis. So we've talked about the pipelines and nothing's really there. We're promoting the use of some sort of structured workflow. But what should we actually keep out? And it all comes down to money at the end of the day. So the EBI Metagenomics is currently just what we store in results files is over 10 terabytes of results. That's without all the sequence information. And so there becomes this balancing act of which intermediates you should store versus reanalyzing the data from scratch. And on the reanalysis, it's not only cost, but it's that reproducibility and the time it's going to take for that reanalysis to happen. So if you've got a reference data set that you want to use as your, this is my ground truth, you don't want to have to keep reanalyzing that data set. So that's really the sort of overview on the sort of standards and best practices that, and you should be thinking about all of those things. The people in red on this slide are the people who've contributed it to that article. Many of them are sitting over there. And if there's any more questions, or if there are any questions from the previous one, I'll take it. Okay.
Okay, so I would like to just ask that, would you then, did I understand correctly that you would rather use raw reads than the assembled context for annotation of metagenomes?
So I would use probably both. So I think assembly has a lot. It depends what you're trying to do. It depends on the question you're trying to ask. I think if you're trying to encapsulate the full functional potential, you would want to look at all the reads. It depends on your community as well and how much sequencing you've done. So if you've not done a lot of sequencing and it's a diverse community, you're not going to get very good assemblies out. And so it's this real balancing game. So I've inherited a pipeline that originally worked on raw reads, and we're about to release a version of that pipeline that can do assembly as well, and we'll have both running side by side, because the amount of data that you ever assemble is never extremely high. There's huge parts of it where it's only, a soil sample is typically 1 to 5% of the data. So if you want to throw away 95% of your data, just focus on the assembly. Could be another way of.
What is the assembler that is at the EBI Metagenomics?
So we use Metaspades as our number one go-to. They're on really, really large data sets, we have to change that. So at the moment, we've just rolling out that pipeline, and so we've just focused on Metaspades as being our primary one. We have two alternatives up our sleeve when we hit those issues, but we're not worrying about those at the moment. We've got it pretty much profiled. We know which ones will work.